Hello and welcome everyone to this podcast on membrane proteins. In the last couple of podcasts, we talked about membrane lipids. And we drew this structure a lot. And we're going to continue to draw this structure a lot. And we spent a lot of time talking about these membrane lipids. We talked about how they could be phospholipids with a phosphate group here. We talked about how they could be glycosidic lipids with a glucose or some other sugar attached by a glycosidic bond. We talked about how they were formed. We talked about how they could be different on one side or the other. Before we add these proteins to this membrane, let's talk about the function of these proteins. Just like the lipids had many different functions, these proteins also have lots of functions. They can serve as receptors or signaling proteins. So let me draw a cell down here, and we could draw the signaling protein like so. And there might be some signal that comes here, interacts with the outside of the cell, and transmits that signal to the inside. There's got to be a way to communicate the outside world of the cell to the inside world of the cell. And these receptor proteins are one of these strategies. The next one we should talk about are structural proteins. And I'll give an example here of spectrin. And what spectrin does is it lines the outside, or rather the inside, of the plasma membrane. It's a cytoskeletal protein that helps provide structure and integrity to the cell. The third function I want to talk about is that of proteins acting as a transporter. A good example of this are the glucose transporters, where glucose, which cannot normally go across the membrane, uses these transport proteins to move glucose into the cell. Next, I want to bring up the example of enzymes. We've of course have talked about enzymes a lot, and we will continue to talk about enzymes a lot, but enzymes can sit right here on the border of this plasma membrane and carry out enzymatic reactions. A good example of this is adenyl cyclase. Adenyl will catalyze the reaction to make cellular CAMP, cyclic AMP. This is a very important signaling molecule in the cell. Let's see, we have a couple more I want to talk about. We have proteins that we're going to put in this electron transport category. These could be proteins in the mitochondria, the chloroplast, or bacteria. Bacteria, they don't have organelles, but these electron transport proteins would be found on the plasma membrane. But let's go ahead and draw a lovely mitochondria here, and these transport proteins will be found on this inner membrane. Another category, I'm going to lump together adhesion and communication. These are going to be found on this cell's edge here, and what they will do is they will allow for the adhesion to a neighboring cell, like so they stick together, and those would be the roles of some tight junctions. However, there are other ones that allow for communication to allow small ions to move from cell to cell, and those would be gap junctions, and I'm just going to put GJ there for gap junction. Now there are of course other functions that we could include here, but I'm going to go and stop here with our list of functions. Okay, now I want to talk about the classes of membrane proteins, and to do so I'm going to make this a little bit bigger so I have some room to work with. The first one I'm going to talk about are transmembrane proteins. As the name implies, trans, it's going to span the membrane. And let's draw a couple examples of a transmembrane protein. So it can be a protein that spans the membrane once, so it can be a single pass or multi-pass. And if it was a multi-pass, we would just have larger structures here. Now sometimes these multi-pass proteins can form a higher ordered structure, allowing channels to form. I'm going to color this one in just for consistency. The next one here is called a monolayer associated protein. And what these will do is they will associate with one part of the membrane, and they may extend, and they probably do extend, into this hydrophobic region, but then they also extend out too. Both transmembrane proteins and monolayer associated proteins are called integral membrane proteins. They are embedded in the membrane at some level. Our next one are lipid-linked proteins. Now what lipid-linked proteins do is they aren't attached directly to the membrane, but they're attached to a lipid component of the membrane. It could be a glycolipid protein and one of the sugars attached to the outside of it. But this lipid-linked protein then is attached to it. So it's associated with the membrane, functions with the membrane, but not directly attached to the membrane. All right, our last one I want to talk about are a class that your book calls protein attached. I often refer to these, and the literature will often refer to these as peripheral proteins. So peripheral proteins will take advantage of one of these integral proteins, like so, or maybe a protein that's not fully across the membrane. And so our peripheral proteins, or the protein attached ones, will bind here, taking advantage of non-covalent interactions. Now we're going to talk about this later, but I think it's a good opportunity to talk about how we're going to remove these proteins if we want to remove them for study. These transmembrane proteins, in order to isolate them from the membranes, they will require a detergent. Triton is a common one used, so is SDS. SDS has the disadvantage of denaturing the proteins, and sometimes we don't want to denature the proteins, but it would certainly dislodge them from the membrane. These monolayer associated proteins would also require a detergent. Detergents come in and they disrupt the membrane, and that's why these are released. Lipid associated proteins require enzymes to remove these proteins from the membrane. So an enzyme like a lipase would work well to release this protein. Now to release these peripheral proteins that are attached to these transmembrane proteins, it requires salt, like sodium chloride, would remove this protein. And we'll see that we take advantage of these features of removing these proteins to identify whether a protein is an integral protein, because it would require detergent to release it, a lipid-linked protein, or a peripheral protein attached to other proteins. Something else that we can do, and we'll come back to this when we talk about a couple problems, is that if we add proteases here to this side of the membrane, not just to this protein, but this whole side of the membrane, we will only degrade these proteins that are here. It won't degrade the whole protein, but it'll degrade some of these proteins. If we apply this protease to this side of the membrane, it's going to degrade all of these proteins that are exposed on this side. It won't get the part stuck in the membrane or this part, but it'll degrade these parts. And that can also tell us a lot about the function of the protein, where it's localized, how much of it is exposed to the outside of the cell, how much of it is left protected in the membrane. These are all important features and tools that we use to study proteins. Now I want to spend some time focusing on transmembrane proteins. And let's go ahead and draw our membrane. And let's draw this transmembrane protein coming through here. And I'm going to try to draw here what should appear as an alpha helix. And I drew this alpha helix because typically an alpha helix is found spanning this membrane. It usually contains 20 to 30 nonpolar amino acids. I say 20 to 30, but I want you to remember this number here, 20. It has to have 20 in order to be able to span here. That is an absolute minimum. When you're looking at a protein and you see 20 nonpolar amino acids in a row, that suggests to you that that is an area that could span the membrane. Doesn't mean it has to, but it could. In previous classes, we talked about alpha helices, and we drew that alpha helical wheel. And we said half of it has to be nonpolar and half of it has to be polar, which might seem a little confusing here based upon the fact I just said we have 20 to 30 nonpolar amino acids. And so I think it's worth spending just a few moments talking about that so that you don't walk away confused. And the reason for that is if we have a folded protein, and I'm going to just draw some beta pleated sheets some regions around here, and then maybe an alpha helix here, and something like this. The reason that in those cases that we were talking about, that it was amphipathic, is that this half of the alpha helix, this half here that's in the middle of the protein, would have to be nonpolar or hydrophobic. I should put that up here too, just so we keep that in mind, that we will often interchange these words, hydrophobic and nonpolar. We mean the same thing. And it needs to be nonpolar because it's interacting with the middle of the protein, and we know that the middle of the protein is nonpolar. And this part here, which is on the outside and perhaps interacting with water, would need to be polar or hydrophilic. So in that context, we were talking about alpha helix in the folded protein. What we're talking about up here is a protein that spans the membrane. And remember, even though I didn't draw them, that within this membrane are all these hydrophobic tails. And as we've said many times, this region here is nonpolar or hydrophobic. So the protein that associates with this region here has to be nonpolar. It's the most energetically favorable way to make this protein work in this membrane. Okay, I think it was worth coming back to talk about that because I didn't want to leave any confusion behind. So let me extend this membrane and then we'll talk about some other features here. We could have a protein that passes through like this and another one over here, and I can't draw it, but there's another one on the back side and another one on the front side, forming this channel. And so each of these would have this alpha helix in it. And in this case, and also remember the one on the back that we can't see would have an alpha helix coming around here, as well as the front side. Now in this case, we could see an amphipathic helix, whereas this side and this side of this membrane, remember we still have membrane here, so these two sides could be nonpolar or hydrophobic, we could say, whereas the inside here would have residues, amino acids, that are polar, creating a nice little channel through here where polar molecules can pass, large polar molecules can pass, such as glucose. Glucose cannot pass through here on its own, but it could pass through one of these channels. So we call this area here that has this polar nature to it, we call this the aqueous pore. People who are studying proteins, they can look at a sequence, they can gaze across the amino acid sequence and make predictions about these membrane proteins. As I've said before, you look for 20 to 30 nonpolar amino acids in a row, that could be a transmembrane protein. Doesn't have to be, but it could be. And so if you have a sequence and you want to know that, you can perform a hydropathy analysis. And what this is just a way to measure long stretches of hydrophobic residues. Over here we're going to have the hydropathy index. Anything above this line is going to indicate hydrophobic amino acids. Anything below it will indicate more hydrophilic. And on the bottom here is the amino acid number. So 1 to say 500. And if what we saw here, something like this, we would study this and look at this and make the prediction that this protein was a multipass protein with two transmembrane, I'm just going to put TM, transmembrane regions. So it might look something like this in the membrane. Again, this just says it could be. It doesn't mean it is. To understand if it is, we'll have to do more studies on this protein, and we'll do that in a little bit. Okay, so let's go to this next step and ask, how are we going to study these proteins? We already talked about the hydropathy index, so we have reason to believe it could be a transmembrane protein. But how are we going to get this protein out? Now, one of the problems we have with transmembrane proteins is that they have this large hydrophobic region. Most studies with proteins are done in an aqueous environment. This protein thrown into an aqueous environment is going to change that protein quite a bit. And so one thing that is done is that detergents are often used. Two common ones are SDS, just like we see in the SDS page gels that we've talked about before. It stands for sodium dodecyl sulfate. And the other detergent often used is Triton X100. SDS is a stronger detergent, and it unfolds the protein in the process. And sometimes we don't want to necessarily unfold the protein. Triton X100 is a milder detergent, and it does not unfold proteins. So how do detergents work then? Well, remember, a detergent is just going to have a single hydrophobic tail with this polar head. And what it will do is, when we mix this with these proteins here, these single hydrophobic units will interact with this protein in such a way that the detergent will surround this hydrophobic region of this membrane protein, attaching to their hydrophobic tails here. They won't attach over here. These will still form hydrogen bonds with the aqueous environment. These detergents will also, in pulling this protein away from this membrane, will interact with the membrane, where the tails of the detergent will interact with the tails of the membrane lipids. In this way, we pull the membrane away from the proteins. Okay, now I want to address, how do we know this is a transmembrane protein and not a peripherally attached membrane protein? And to do this, I'm going to draw a peripherally attached protein attached to this here. And we're going to take advantage of the fact that we know that salt will release the peripherally attached protein. And to remove the transmembrane protein, we'll need a detergent like Triton X. What you'll do first is you'll take this membrane sample here, and you will spin it in a centrifuge, in which case all the membranes will end up in the pellet. And the aqueous environment that this membrane was in will be here in the supernate. And to remind you, in case you haven't had it before or recently, we use these terms supernate and pellet. Pellet is just the part that is the heaviest that spins to the bottom of the tube, and the supernatant is what is at the top, the liquid portion. Now we could run a SDS page gel here, and if we're looking at these two proteins here, we'll still see both proteins. And just for argument's sake, let's say that this protein is 50 kilodaltons and this transmembrane protein is 75 kilodaltons. So we should see both of these proteins, 75 and 50. And remember, this is our membrane fraction here. Now, what we do next is we take this membrane fraction and we treat it with salt. And we spin this reaction again, and we get a pellet and a supernate. And we run an SDS page on the supernate and the pellet. So here we're going to put the supernate, and here we'll have the pellet. What the salt will do is it will release this peripherally attached protein. And so it will be in the supernate. So I'm just going to do this to color coordinate with this protein here. So it's going to be suspended in the supernate. And so when we spin this down and we run the supernate on an SDS page gel, you'll see this 50 kilodalton protein. You won't see the 75 kilodalton protein in the supernate, but you will see it in the pellet because it's still attached to the membrane. And the membrane, being this heavier component, will pellet. So we'll still see it here. The next thing we're going to do is treat it with our detergent, Triton X100. Triton X100 is going to disturb the membrane and the protein around here and cause this protein to dissociate from the membrane. So again, we spin it in a centrifuge. We have our pellet here, and we have our supernate. The detergent is going to be strong enough to have released this protein, and so now it's going to be in the supernate. It will be in solution in the supernate, but it'll be in the supernate. The membranes that's left over will still be in the pellet. So again, we will run our gel like so, and we'll look at our supernate and our membrane. In our supernate, up here, we will see our 75 kilodalton protein. This one here, the transmembrane protein. In the pellet fraction, we won't see anything. We won't see our two proteins. So this is what we would see based upon the drawing I just showed here. The question I have for you is, what if you did this experiment and you of course didn't know what you saw up here, what you see up here, because you're doing the experiment. You don't know where this protein is attached. So let's say you did this experiment, and after the Trident X100, you saw this instead of this. We saw that 75 kilodalton protein still in the pellet and not in the supernate. What could be going on here? Why is it still associated with the membrane? Come up with an idea of what could be happening here and how you would go about showing the nature of this protein. I'm not going to tell you right now. We'll talk about it in class, but I bet you can figure it out. Okay, I'm going to change gears quite a bit here, still talking about cell proteins, but moving away from those transmembrane proteins. And I want to talk about the cell cortex. So here we have our cell, and as I drew on the opening slide, we know that there are proteins all along this edge that help provide structure and support for the cell. Helps maintain its integrity, helps determine its shape, does a lot of things. And so this layer of proteins here is what we refer to as the cell cortex. It's rich in cytoskeletal proteins. These proteins can help anchor membrane proteins to help prevent some of their free movement in the membrane. Now what I want to do is show a different view here, like as if you were inside the cell looking out to the periphery here. So I'm going to draw it as a rectangle here, showing black here to kind of show you still the outline of the cell where the plasma membrane would be. But really the plasma membrane is going to be all this area here. Because remember, we're looking this way. And if we're looking this way, what we see is this very structured arrangement of proteins. It forms this almost skeletal-like structure that provides strong network to give that cell, as I said, its shape and its integrity. Protein structures here I've drawn here are an example of something called spectrin, which is this protein right here. The importance of spectrin and maintaining this cell cortex is seen if we look at red blood cells. And I'll try to draw a red blood cell as best as I can, but red blood cells are very flexible. They need to have this kind of this elasticity to them so they can move through very small openings in our circulatory system, like capillary vessels. If spectrin is mutated, so I'll put a plus here because that's the non-mutated one. If it's mutated, the red blood cells, instead of forming this concave flexible structure, they form these spherical, much more rigid structures shown here. These lead to anemia. They're not able to deliver oxygen more effectively. There's fewer red blood cells in this case, and they're fragile because they lack that flexibility, because they lack this spectrin that is helping to maintain the functionality of this red blood cell. All right, one other thing I want to mention about spectrin is that they will serve to anchor various proteins. So if I had a protein here, so maybe a transmembrane protein, or maybe a protein here, they would serve to hold that protein in place. Now, not all the proteins are going to be held in place like that, but some might. And they do that by taking these proteins, these transmembrane proteins or peripherally attached proteins, and holding them there with these attachment proteins. So what I've drawn there, let me draw it out, write it out here, attachment proteins. So attachment proteins bind to the spectrin and then anchor these transmembrane proteins or the peripheral proteins, but whatever protein might be associated with the plasma membrane here. Next, I want to talk about protein movement within the membrane. Now, in general, we can think of proteins in the membrane, let me draw some over here not to confuse them with those, as having the capability of moving freely. Now, some of the things that could restrict their movements could be these attachment proteins, shown here, but another thing that could restrict their movement involves these structures called tight junctions. And I'm going to erase this here so I can draw a different picture. So the question is, how do we keep these proteins from moving over here? Let's say for some reason we want to make sure they don't move in this direction. We need to have some kind of block here, some physical block. And the reason this might be important is the same reason that membrane lipids are sometimes found in certain regions and not other regions. Same thing with proteins. It could be that this side of the cell has a specific function for these proteins. So we need to keep these proteins over here. We don't want them over here. These proteins here have a specific function on this side. So we want to be able to restrict movement to some extent. To illustrate this, I want to draw some epithelial cells. These could be the lining of our stomach. That would make sense. And let's draw some proteins here. And these orange proteins slowly are looking red mixed with the black, but also call them orange. We want to keep on this lower 2/3 of the cell. And then we have these other proteins, and we'll make them blue, that we want to keep in this upper third or so of the cell. The blue proteins have some kind of function that is important in this part of the tissue. These orange proteins have a function down here. For the purpose of this discussion, it doesn't really matter what that purpose is. We just know we have this differing function. And what happens is that these cells are held together by a protein complex called tight junctions. These tight junctions we'll talk about later in the semester in a little bit more detail. But for right now, what you should know is that they prevent the movement of these proteins from moving down here. They also prevent the orange proteins from moving up the space. In addition, and this is something we'll talk about later, is that they also prevent the mixing of these two environments. So if, just to add something here, if we had an anchor protein here holding on to some structure in the cell, some other macromolecule structure or something, not sure what, doesn't matter so much, these structures here would also have their movement limited too. I want to just spend just a tiny bit of time on glycoproteins. In the previous podcast, we had talked about membrane lipids. And we said that membrane lipids could have sugars attached to them, as I'll show here with the G's. Maybe not on both sides, probably not on both sides, but they would have them attached. Glycolipids is what these were called. But we also know that we have proteins here. And we'll just draw our familiar friend here, the transmembrane protein, and we're going to attach a sugar to it. Adding these sugars to these structures is a process that we call glycosylation. So a glycoprotein is any protein like this that has attached to it some kind of sugar using a glycosidic bond. So what are the functions of these? Lots of functions, but we'll list a few. The first is their role in cell-to-cell recognition. Certain sugar patterns here will help the immune system recognize self cells versus foreign cells. They're also important in receptor ligand binding. Certain proteins will recognize this, and that's how they will associate with the membrane. The last one I want to mention is that this glycosylation protects cells from being digested by some foreign invader. And as I'm looking at this, I forgot to say one thing that I need to add real quick, and that glycosylation occurs in the ER. And each glycosylation is enzymatic specific. There's a different enzyme needed to add different kinds of sugar attachments. Okay, that ends what I want to say for membrane proteins. Over these past two podcasts, we've talked about the functions of membrane proteins. We talked about classes of membrane proteins. Were they peripheral, were they integral, were they lipid linked? We talked about ways we could remove those proteins in a specific way to determine the nature of the proteins. We talked about the cell cortex and the importance of the cell cortex in arranging protein interactions. We talked a little bit about protein movement and how we can restrict protein movement. And finally, we ended with glycoproteins and their importance within a cell and within an organism. Okay, if you have any questions at all, please make sure you come see me. If not, I'll see you in class. Bye.